There are 4 types of main biological molecules, which are carbohydrates, lipids, nucleic acids, and protein. For practical one, we conduct experiment on 3 biological molecules, which are carbohydrates, proteins, and lipids. As for carbohydrate, it consists of carbon, hydrogen, and oxygen in its chemical structure. The basic tests frequently used to test for certain carbohydrates are Mollis test, general carbohydrate test, Borfoit test, monosaccharide, Benedict test, reducing sugar, and polysaccharide. Proteins are also composed of carbon, hydrogen, nitrogen, oxygen, and sulfur atoms. The basic tests frequently used to test for certain proteins are burette test and Milan test. Lipids, which are composed of carbon, hydrogen, and oxygen atoms. Lipid is an organic compound that consists of carbon, hydrogen, and some oxygen. The basic tests frequently used to test for certain lipids are acrolein test and coloring test.
Measure 2 ml for each sucrose 2%, glucose 2%, maltose 2%, and starch 5% in 4 different test tubes. Add 5 drops of alpha-naphtha solution 5%. Mix by gently shaking. Slowly place 1 ml concentrated sulfuric acid into each of these test tubes. Make sure do not shake them. Observe the color change and record the result of the experiment.
Add 2 ml of sucrose, 2%, glucose 2%, maltose 2%, and starch 5% into each test tube separately. Then 5 ml barfoot tester into sucrose 2%, 5 ml barfoot tester into glucose 2%, maltose 2%, and starch 5%. Soak them for 30 seconds in the boiled water.
Place 1 ml of sucrose 2%, glucose 2%, maltose 2%, and starch 5% into each test tube separately. Add 2 ml Benedict solution into each test tube. Mix by gently shaking. Then heat briefly in boiling water for 2 minutes. Cooling them down before you observe any changes and record the result. The solution of sucrose and starch remains blue, while glucose and maltose change from clear blue to brick red color.
Place 5 drops of sucrose 2%, glucose 2%, maltose 2%, and starch 5% onto tile patches. Add 1 to 2 drops of iodine potassium iodide solution and observe.
Place 3 ml albumin solution 5% and milk into 2 different test tubes. Add 3 ml sodium hydroxide 2% and mix gently. Add 4 drop cuprum sulfate 0.5% and observe the color changes and record the result.
Place 3 ml albumin solution 5% into test tubes. Then, place 3 ml milk into test tubes. Next, add 3 drops of nylon solution and then observe and record the result.
Place 5 drops of coconut oil, palm oil and vegetable oil into 3 different test tubes. Add 1 gram sodium bisulfate and heat slowly using Bunsen burner. Indicate the smell present and record the result. The smell present for coconut oil gives pungent smell. The smell present for vegetable oil does not give pungent smell. The smell present for palm oil does not give pungent smell.
Place 3 drops of coconut oil, palm oil and vegetable oil into 3 different test tubes. Add 5 ml of distilled water and mix by gently shaking. Add a few drops of Sudan 5. Carefully mix by hard shaking. Leave them for 1 minute and observe.